Hello everyone, this is Dr. Alvin Singh. So now we are going to discuss the topic of chloroplast, right? The third type of plastic. Already in the previous video guys must have seen about leucoplast and chromoplast. This is the third one, chloroplast. So as I told you that the word chloro, right, which is related to the pigment of color of green, right? So this chloroplast available in plant cells. This one I told you at the beginning slides also in with the introduction of plastic. Yes, available in plant cell, algae, some few protists also having this chloroplast. Example I have given a euglena over here, right? So second one is chloroplast, shape of chloroplast. Generally they are in a biconvex shape, right? Biconvex. Simply I can show the diagram over here. Biconvex shape will be there. That is it. So it is available in the higher plants. Apart from that, the structure, the shape will be available in a different form. They will be in a filamentous form, saucer shaped, ovoid, discoid, club shaped, ribbon shaped, right? Ribbon shape also I can write here. Right, in Spirogyra you must have seen that ribbon shape available. So different shape of chloroplast available in this. So what will be the size? The size seriously varied between species to species. Example, one of the large sized in the sun I can say from the case of Chlamydomonas. Same way, any species, as I have given examples over here, even in within a plant cell also the size getting different from species to species. That's what I have written the word first point like this. Second one is commonly the thickness of chloroplast 2 to 3 mm in thickness, diameter 5 to 10 mm in diameter. When you are discussing about the structure, you come to know about this. So how many numbers of, as I told you on the earlier slides, you must have seen that the numbers also will be varied. So here I see as the same way only I have written there, single large chloroplast available in the Chlamydomonas. In Chlamydomonas structure, I hope a simple structure, let me draw here. Right guys? Right? So this is completely, yes. Apart from this structure only, we can find out your other cell organelle, right? Nucleus, mitochondria, this other cell organelle will be available. But maximum space is occupied by chloroplast in Chlamydomonas. Then the maximum number, mesophyll cells, which are they are containing maximum chloroplast. The number is 20 to 40 chloroplast in the mesophyll cells. I hope you all know that where is mesophyll cells, right? In leaf structure, you must have seen that the upper layer and lower layer, dorso-ventral region. Between that, whatever the cells which are available, that all comes under mesophyll cells. Okay, guys. There, each cells are having 20 to 40 chloroplast over there. So let us discuss about their structure in the next slide. Okay. So now we have a structure of chloroplast here. See now, look at that. This is the nature. So let us label one by one. This is outer membrane. This is inner membrane. This is, right, space, the intermembrane space. Right, we can write that space of intermembrane. Okay guys, this is the major part of this. Right, so now let us enter inside, because this point also important, because from the beginning onwards I used to say that double membranous structures, right? So plastids are, I told you that double membrane. So like the same way only I have given here outer and inner membrane over here. So when you are coming over inside, so this is, I mean inside, the particular portion is called stroma. Look at the word carefully. This is stroma. Because why I am telling this, in biology we have crossed in a same plant nature, stoma. The word stoma we crossed already, right? It is a stomatal opening will be called by the word stoma. S-T-O-M-A. Here stroma. Right. So now the each coin arrangement, like the slack of coin arrangement which is available. Individually, if I want to call it, this is called thylakoid. Individually, if I want to call it, its name is thylakoid. If we want to call the particular stack of the coin-like structure, the arrangement of coin like this, we want to call it together, so I have to use the term granum. Okay, so the plural form of granum. I mean, if I want to collectively call all the granum, then I have to use the word grana. Plural form is grana. Plural form. Period stands for plural here. Okay, fine. Now, apart from that, see this area. So the granule, each granum is connected by this. This is called by the word stromal lamellae. Okay, whatever the structure is, we can call it stromal lamellae. Otherwise, look at the word intergranal lamellae also. Intergranal, because between the granule it is available, no? So intergranal lamellae over there. Apart from the word, the first slide you must have seen that chloroplasts, especially plastids, are semi-autonomous, I said. It means what? They have their own DNA, own ribosomes like this. So we can label it, yes, they have a DNA inside. If you can consider this is the ribosome, that is 70S ribosome, right? Available on this. Yes, and then the red color which I have given over here, that is starch storage, starch grains which are available over there. These are the structural arrangement of this. Now we are going to discuss these points one by one. Example, if I want to talk about the membrane, the envelope size is almost 50 to 100 angstrom in size. The same way, the space which is available between the membrane, that is 100 to 200 angstrom in size. That is the size of this, the space which is available on this, these all. Now in the next slide, let us talk about the internal portions one by one. How the grana arrange, how the stromal arrange, everything on this, right? So, stroma, it is the portions where the matrix, ground matrix is available, that is called stroma, I said. That stroma generally in a gel fluid like structure, jelly like structures over there. This stroma which contains 50% of protein of the total chloroplast. Example, proteins generally available over there. In that, only stroma alone, for example, protein available in a thylakoid, protein available in a membrane. Compare with that, only this region, stroma alone holding 50% of the chloroplast. And then stroma, this is the portion actually containing that particular ribosomes and DNA structure. I told you at the beginning also, because of this presence only, they are semi-autonomous in nature, I told you that. Right, guys? So it means they can control their own functions, own structure like this. So it means as they have a ribosome and DNA, yes, what is the result? Yes, they can produce their own protein. Right? Mainly that protein which is used for their structural arrangement. They are called structural protein. So the semi-autonomous, their own transcription, translation, they will produce their own structural protein. This is the information about stroma. And then this stroma is the place for the particular process in the light reaction, dot reaction we will study in future in a photosynthesis step. So this is the place for a dark reaction. So once the light reaction gets over, dark reaction will be held at this place only. Place for dark reaction. That is also we will study that later. So I am coming for the next one. That is thylakoid, the arrangement. As you have seen that before in a structure, right? How thylakoids are. So they are the flattened sac-like structures, right? So the particular group of the sac-like structure is known as grana, right? So it means how many grana possibly in one chloroplast. It means what? Example, look at that. In a chloroplast, the coin arrangement which are available like this, correct guys? So like this, for example, I have given here 3 grana over here. Like this, if you are looking at one chloroplast, we can find almost 40 to 60 grana in the matrix. Then, second one is that each granum the number of thylakoids will be 1 to 100, right? Understand that? This structure, right? This is the coin arrangement as I told you, no. So how many? Here I have given 1, 2, 3, 4 like this. So like this, if you are looking at one single granum can contain 1 to 100 thylakoids over there, right? So now let us talk about their pigments arrangement.